In the sedimentary layers of Gale Crater, which have been explored for more than a decade by the Curiosity rover operated by NASA, a chemical anomaly has been identified whose significance extends far beyond routine reports of organic detections on Mars. This is not merely the presence of carbon-bearing compounds, but the concentration and molecular length of hydrocarbons that, according to calculations by NASA research teams and collaborators, exceed the expected limits of standard abiotic synthesis mechanisms. Within a sample of ancient mudstone formed in a lacustrine environment billions of years ago, the sample analysis at Mars instrument aboard Curiosity detected decane, undecane, and dodecane. These are saturated hydrocarbons with carbon chains consisting of 10 to 12 atoms. In planetary geochemistry, these are non-trivial molecules because the longer the carbon chain, the less likely it is to accumulate and persist without specialized chemical conditions. Usually, we expect to see simple, short-chain organics, the kind that can be easily assembled by random chemical processes or delivered by meteorites. But seeing chains of 10, 11, and 12 carbon atoms in these specific concentrations forces us to pause and reconsider our models. NASA scientists emphasize that such compounds are not by themselves biosignatures. We must be very clear about that from the start. Finding a long carbon chain does not inherently mean we have found life. However, their preservation and the reconstructed original concentrations create a statistically meaningful deviation from purely abiotic expectations. When we look at standard models of how non-biological chemistry works on Mars, we expect a certain distribution of molecules. We expect a drop-off in abundance as the molecules get larger and more complex. What we are seeing here is a profile that does not quite fit that smooth curve. It suggests that something else was going on, something that favored the production or the preservation of these specific lengths. This is the heart of the anomaly. It is a puzzle piece that does not fit the picture we have been building of Martian geochemistry. To understand why this is so strange, we have to look at the environment where these molecules were found. This is ancient mudstone, rock that started as mud at the bottom of a lake billions of years ago. Over time, that lake dried up, the mud hardened into stone, and it sat there on the surface of Mars. And this is where the story gets complicated. Identifying these specific hydrocarbons, decane, undecane, and dodecane, raises immediate questions about their survival. Simple organic molecules are tough. They can survive a lot of abuse. But a chain of 12 carbon atoms is fragile. It can be broken. Thermal energy, radiation, chemical oxidation, all of these forces work to snap those chains back into smaller pieces. The fact that the sample analysis at Mars instrument is seeing them now, after all this time, tells us something about their history. It implies that either they were protected in a very specific way, or there were just so many of them to start with that even after billions of years of degradation, we still have enough left to detect. This finding challenges our baseline assumptions. When we model the organic inventory of Mars, we usually input the known rates of production from things like volcanic activity or chemical reactions between water and rock. We add in the amount of material delivered by comets and asteroids. Then we subtract the material destroyed by radiation and harsh surface conditions. When the NASA team runs those equations for Gale Crater, the numbers for these specific long-chain hydrocarbons do not balance. There is a surplus. There is more decane, undecane, and dodecane than the equation says there should be. This discrepancy is what makes this finding so compelling. It is not just about finding a needle in a haystack. It is about finding a pile of needles where there should be none. If you are following the developments in planetary science, you know that we are always looking for patterns. Life, as we know it, loves patterns. It builds specific molecules for specific jobs, creating concentrations of certain compounds while ignoring others. Abiotic chemistry, chemistry without life, tends to be messier, more random. It creates a little bit of everything. The profile of hydrocarbons in this mudstone looks less like random noise and more like a specific signal. It is a signal that has been degraded over billions of years, faded by time and radiation, but it is still recognizable as distinct from the background noise. This is why the detection of these three specific molecules is generating such intense interest. It pushes the boundaries of what we thought was possible for the preservation of complex organics on the surface of Mars. We need to consider the implications of this discovery carefully. It suggests that the ancient lake environment in Gale Crater was chemically complex. It was not just a basin of water. It was a dynamic chemical system capable of concentrating and preserving larger organic molecules. Whether that complexity came from an unusual geologic process we have not yet identified, or whether it hints at something more profound, is the question that is now driving the next phase of analysis. The presence of decane, undecane, and dodecane is a data point that demands an explanation. It is a stubborn fact that refuses to be smoothed over by our current understanding of the red planet. If you appreciate deep dives into this kind of complex scientific data, make sure you are subscribed to the channel so you do not miss our future breakdowns of these findings. We will be tracking this story as new papers are published and as the Curiosity team releases further analyses. This anomaly in Gale Crater is a window into the deep past of Mars. It is a glimpse of a time when the planet was wetter, warmer, and chemically active. And in that ancient environment, something created or concentrated these long hydrocarbon chains. As we move forward, we are going to look at the factors that seemingly should have destroyed these molecules long ago. We need to talk about radiation, about time, and about the incredible odds against these molecular chains surviving to be detected by us today. Because when you do the math on their survival, the mystery only deepens. The fact that they are here at all is, in itself, a remarkable defiance of probability. As we established in the previous analysis, the detection of decane, undecane, and dodecane in the ancient mudstones of Gale Crater presents a significant chemical puzzle. But to truly grasp the magnitude of this anomaly, we must look at the hostile environment these molecules have endured. A central component of the analysis involved reconstructing the radiation history of the sample. Unlike Earth, Mars lacks a global magnetic field to deflect charged particles from space. It also lacks a dense atmosphere to absorb high-energy radiation. This leaves its surface exposed to a continuous, relentless bombardment by galactic cosmic rays. These are not just light waves. They are high-speed atomic nuclei that smash into the Martian surface with incredible energy. When they strike rock, they shatter atoms and chemical bonds alike. Geological assessments from the NASA mission team estimate that the studied rock layer has been exposed for roughly 80 million years. That is 80 million years of sitting directly in the line of fire of cosmic radiation. On a planetary timescale, 80 million years might sound like a blink of an eye, but for an organic molecule, it is an eternity of destruction. Over such timescales, ionizing radiation should significantly degrade complex organic molecules. Imagine a long chain of carbon atoms, like the 12-carbon chain of Dodecane. Every cosmic ray that hits near it, every secondary particle cascade that passes through the rock, carries enough energy to snap those bonds. It is like trying to keep a delicate glass sculpture intact while it is being pelted with hail for millions of years. Laboratory irradiation experiments and computational simulations, supported by NASA, indicate that long-chain hydrocarbons gradually fragment into shorter compounds and simple gases under these conditions. If you start with a long chain, radiation will chop it up. You get shorter chains, then smaller fragments, and eventually you are left with simple gases like methane or carbon dioxide that escape into the atmosphere. The process is unidirectional. Radiation destroys complexity. It does not build it. Therefore, the mere detection today of trace amounts of 10 to 12 carbon atom molecules implies that their initial abundance must have been considerably higher. This is where the math gets incredibly interesting. If we see a certain amount of decane today, and we know that 99% of it should have been destroyed over the last 80 million years, then the amount that was originally there must have been enormous. We are looking at the survivors of a chemical massacre. The reconstruction of that original concentration is what creates the statistical deviation. Standard abiotic models can account for some organics. They can explain a certain baseline level of carbon chemistry. But when you take the current detection and backtrack through 80 million years of radiation damage, the starting number you arrive at is far too high for those standard models to explain. It is not just about the surface exposure either. Galactic cosmic rays can penetrate meters into the rock. There is no hiding place in the upper layers of the Martian surface. The radiation creates a cascade of secondary particles, neutrons, and gamma rays that scatter through the subsurface, continuing the work of breaking down complex molecules. The NASA team has modeled this flux with high precision. They know the density of the mudstone. They know the energy of the incoming rays. The degradation rates are well constrained by physics. This is why the finding is so robust. It is not based on a guess about how stable these molecules are. It is based on hard physics and experimental data on how hydrocarbons behave under irradiation. The anomaly, therefore, is a quantitative one. It is a problem of mass balance. Where did this massive initial reservoir of long-chain hydrocarbons come from? If the radiation history is correct and the degradation models are accurate, then the ancient lake in Gale Crater must have been rich in these specific compounds, much richer than we dared to imagine. This forces us to look for a source that is capable of producing not just trace amounts, but significant concentrations of 10, 11, and 12 carbon chains. We have to ask ourselves, what kind of environment could produce such an abundance, and how could it produce it specifically in these chain lengths? Random processes do not typically favor such specific outcomes in such high quantities. If you are rolling dice chemically, you get a bell curve. You get a lot of small stuff and very little big stuff. But here, the survivors tell us that the big stuff was surprisingly common. As we consider this radiation history, the mystery shifts from simple detection to a question of origin and preservation. It is possible that the molecules were protected for a long time before being exposed. Perhaps they were buried deep underground, shielded by hundreds of meters of rock, and only uncovered by erosion in the last 80 million years. That is the timeline the geologists are working with. But even with that burial, the rock still accumulates damage. Radioactive elements within the rock itself, uranium, thorium, potassium, decay and release radiation from the inside out. The NASA models account for this internal radiation as well. Even deep underground, these molecules were not perfectly safe. They were slowly being cooked by the natural radioactivity of the crust. So, whether we look at the external bombardment from space or the internal decay from the rock, the conclusion remains the same. The molecules we see today are the remnants of a much larger population, and that population was chemically distinct, dominated by these Decane, undecane, and Dodecane chains. This points to a production mechanism that was efficient, sustained, and capable of generating complexity that contradicts our standard abiotic assumptions. This deepens the intrigue significantly. If the radiation should have wiped these slate clean, and it did not, then the writing on that slate was originally written in very bold, heavy ink. In the next chapter, we will look at the usual suspects for this kind of ink, meteorites, and geological chemistry, and see why they also fall short of explaining what we have found. Building on this understanding of the radiation environment, we have to turn our attention to the sources that usually supply Mars with organics. We have a good catalog of what falls to Earth and what cooks out of the ground. But do any of those match the fingerprint we are seeing in Gale Crater? That is the question we tackle next. But before we move on, I want to hear from you. What do you make of this survival paradox? Do you think we are underestimating the resilience of these molecules? Or are we underestimating the ancient Martian environment? Leave a comment below with your thoughts. Given what we now understand about the radiation history and the massive initial abundance required to explain the current detection of decane, undecane, and dodecane, we must critically evaluate where these hydrocarbons could have come from. In planetary science, whenever we find organics, we have a lineup of usual suspects. We look at sources that we know exist and that we know can deliver carbon to Mars. The primary suspect is always meteoritic delivery. Mars, like Earth, is constantly bombarded by debris from space. Dust, meteorites, and comets rain down on the planet, bringing with them a cargo of organic compounds. Models of meteoritic delivery of organic material, extensively studied within NASA's planetary science programs, estimate the flux of carbon-bearing compounds to the Martian surface. We have a very good handle on this. We analyze meteorites that fall to Earth. We analyze the dust in the upper atmosphere. We know the chemical recipe of the solar system. Carbonaceous chondrites, a common type of meteorite, are loaded with organics. They contain amino acids, hydrocarbons, and all sorts of complex chemistry. So, the first question the NASA team asked was, Could this just be ancient meteorite dust accumulated in the lakebed? Even accounting for intense bombardment in the planet's early history, the calculated influx appears insufficient to explain the reconstructed concentrations in this specific stratigraphic horizon of Gale Crater. The math just does not add up. To get the concentration of C10, C11, and C12 hydrocarbons we are seeing, you would need a rain of meteorites so dense and so specific in its composition that it defies what we know about the early solar system. Meteoritic organics are a complex mix. They are a soup of everything. They do not typically produce a sharp, distinct signal of just these specific saturated hydrocarbons in these quantities without a lot of other background noise that is missing here. Furthermore, the delivery process itself is violent. Impactors hitting the atmosphere or the ground generate immense heat. This heat destroys a lot of the delicate organics before they can even settle into the sediment. While some material survives, the efficiency of delivery is low. When you compare the influx rates with the preservation rates we discussed in the previous section, the meteorite hypothesis starts to crumble. It can explain some carbon, yes, but it cannot explain this carbon in these amounts in this specific rock layer. So, if it did not come from above, could it have come from below? We turn to the second usual suspect, geochemistry. We know that rocks and water can make organic molecules. Processes like serpentinization, where water reacts with iron-rich rocks, can release hydrogen, which then reacts with carbon to form hydrocarbons. This is known as Fischer-Tropsch type synthesis. It happens on Earth in hydrothermal vents. It happens in industrial chemical plants. It is a powerful mechanism. Known geochemical pathways, including serpentinization and catalytic synthesis on mineral surfaces, have been rigorously considered by the team. Yet, under established conditions, these processes rarely yield sustained accumulations of saturated chains of this length in significant quantities. Abiotic synthesis tends to make a lot of methane, 1 carbon atom, and ethane, 2 carbon atoms. As you try to build longer chains, the efficiency drops off exponentially. Making a 10-carbon chain strictly through geology is hard. Making enough of it to saturate a mudstone layer is even harder. The thermodynamics just do not favor it. You need very specific catalysts, very specific temperatures, and very specific pressures. And even then, you usually get a broad distribution of molecules, not a cluster around the C10 to C12 range. The detected anomaly implies a selectivity that rock chemistry does not usually possess. Rocks are indifferent. They do not care if they make a 9-carbon chain or a 13-carbon chain. Biology, or perhaps some exotic prebiotic chemistry, is picky. It selects specific lengths for specific functions. The signal in Gale Crater looks picky. It looks like something was driving the chemistry toward these specific molecular weights. This is where the standard abiotic models hit a wall. We have ruled out the sky, and we have ruled out standard rock chemistry. Neither source provides a flux high enough or a composition specific enough to match the data from curiosity. This leaves us with a gap between what our models predict and what the rover is actually seeing. And in science, gaps are where the most interesting discoveries are hiding. The fact that known non-biological mechanisms imply a deficit suggests we are missing a mechanism. Could there be a localized geological source we have never seen before? Perhaps. Maybe there are catalytic clays in Gale Crater that act like super-efficient chemical factories. But implying a new, unknown geological process is just as significant as implying biology. It means the dead red planet was doing some very sophisticated chemistry in its youth. This brings us to a tantalizing possibility. If the hydrocarbons are not the primary product, if they are not what was originally made, then what are they? Further intrigue arises from the possibility that the detected hydrocarbons are thermal breakdown products of more complex carboxylic acids. This connects to what we see in the oil and gas industry on Earth. You start with complex biological lipids, you heat them up over millions of years, and they crack into smaller, simpler hydrocarbons. Decane, undecane, and Dodecane are classic cracking products. They are what you get when you break down fatty acids. This shifts the conversation completely. We are no longer just asking, how did we make these hydrocarbons? We are asking, what bigger molecule did these hydrocarbons come from? And that question leads us directly into the realm of biochemistry and the structures of life itself. If these are indeed fragments of something larger, then the original anomaly, the needle in the haystack, was even bigger and more complex than what we are seeing today. We are looking at the shattered remains of a much grander structure. In the next part of our deep dive, we are going to explore this connection to fatty acids and lipids. We will look at why finding the wreckage of these specific molecules is so provocative and how it mirrors the signatures of ancient life found on Earth. This is where the chemistry starts to look remarkably familiar to those of us who study the machinery of cells. If you are enjoying this rigorous breakdown of the data, please take a moment to like the video. It helps us get this science out to more people. Now, let's look at the ghost of the molecules that might have been there before the radiation took its toll. We are going to talk about lipids. This connects to one of the most intriguing hypotheses emerging from the Gale Crater data. As we have seen, the direct abiotic synthesis of decane, undecane, and dodecane in high concentrations is difficult to explain with standard models. But what if these molecules are not the original actors in this story? What if they are merely the debris left behind by something far more complex? The analysis points towards the possibility that these hydrocarbons are thermal breakdown products of long-chain fatty acids. On Earth, long-chain fatty acids are fundamental components of lipids. These lipids are the building blocks of cellular membranes. Every cell, from the simplest bacteria to the most complex human tissue, is wrapped in a membrane made of these compounds. They are the barrier between life and the outside world. They keep the chaotic chemistry of the environment out and the organized chemistry of life in. The carbon chains in these membrane lipids often fall into specific length ranges, and when they degrade over geological timescales, when they are heated and pressed into rock, they break down into saturated hydrocarbons like the ones Curiosity has found. NASA researchers stress that abiotic synthesis of such acids is theoretically possible. We can make fatty acids in a lab without life. It is possible. But, and this is a crucial distinction, abiotic synthesis typically favors shorter chains and requires particular catalytic environments. It does not naturally churn out the precise abundance of the specific long chains required to produce the signal we see in Gale Crater. The fingerprint of abiotic fatty acids is usually distinct from the fingerprint of biological ones. Biological systems are ruthlessly efficient. They make exactly what they need, say a 16-carbon or 18-carbon chain, and they make tons of it. They do not make a random smear of chain lengths. When those biological lipids degrade, they leave behind a ghost of that preference. The breakdown products, the hydrocarbons we see today, would reflect that original specificity. The presence of C10, C11, and C12 hydrocarbons could be the result of the cracking of these larger specific biological precursors. If ancient microbial ecosystems once existed in the lakes of Gale Crater, lipid-like compounds could have accumulated in bottom sediments. Imagine a lake teeming with microscopic life. When those microbes died, they sank to the bottom. Their fragile proteins and DNA would have been destroyed quickly, but their membranes, tough, waxy, lipid-rich membranes, are much more resistant. These lipids could have been buried in the mud, trapped within the mineral matrix. Over time, the mud turned to stone. The minerals around them would have acted as a shield, protecting them from the immediate effects of oxidation and ultraviolet degradation. But deep underground, heat and time would work on them. The long lipid chains would snap, breaking down into the stable hydrocarbons we detect today. This process, known as diagenesis, is exactly how oil deposits form on Earth. We are essentially describing the potential precursors to a Martian fossil fuel, albeit on a microscopic scale. The strength of this hypothesis lies in how well it fits the data. It explains the concentration. Biological communities can produce massive biomass. It explains the specificity. Biology selects for specific chain lengths. And it speaks to the preservation paradox. Lipids are among the most durable biomarkers we know. If you were going to design a molecule to survive 3 billion years and tell a story about its origin, a lipid breakdown product is a very good candidate. However, we must be scientifically rigorous. NASA frames its conclusion cautiously. None of the examined non-biological mechanisms fully accounts for the observed organic abundance. This phrasing is precise. It does not say it is aliens. It says physics and chemistry, as we currently understand them on Mars, are missing a variable. That variable could be a biological origin, or it could be a prebiotic process that mimics the selectivity of life. Perhaps there is a way for mineral surfaces to act as templates, organizing carbon atoms into these specific long chains without a living cell being involved. That defines the frontier of prebiotic chemistry, the blurry line between dead matter and living matter. If the lipid connection holds true, it implies that the ancient lake in Gale Crater had the ingredients for cell membranes. It had the boundary layer necessary to compartmentalize life. Even if these molecules were made abiotically, their presence means the environment was capable of self-organization. You cannot have a cell without a membrane. You cannot have a distinct individual organism without a way to separate self from environment. The presence of these specific hydrocarbon chains suggests that the chemical potential for that separation existed. We are looking at the raw materials of individuality, Whether they were ever assembled into a living, dividing cell is the ultimate question. But the discovery proves that the Lego bricks for building a cell wall were scattered on the floor of Gale Crater, and there were a lot of them. This leads us to the final part of our analysis. We have an anomaly. We have a list of suspects that don't fit, and we have a provocative link to the machinery of life. So, where does NASA go from here? How do we move from anomalous data to a definitive answer? The implications of this finding are forcing a shift in how we plan future missions and how we interpret the history of our planetary neighbor. This is not just about looking back, it is about deciding where to look next. In the final chapter, we will discuss the paradigm shift this data represents and what it means for the future of Martian exploration. We are standing on the edge of a new understanding, and the next steps are critical. Thus, NASA frames its conclusion cautiously. None of the examined non-biological mechanisms fully accounts for the observed organic abundance. This statement, while understated in the typical language of scientific reports, represents a seismic shift in our perspective. This is not a confirmation of life, but rather the identification of a discrepancy between empirical data and theoretical expectations. In the history of science, such discrepancies often mark the beginning of paradigm refinement. We saw it when the orbit of Mercury did not quite fit Newton's laws, leading to Einstein's relativity. We are seeing it now on Mars, where the chemical inventory does not quite fit our abiotic models. The detection of these anomalous hydrocarbon concentrations, decane, undecane, and dodecane, is a forcing function. It forces us to acknowledge that our simulations of the Martian past are incomplete. We are missing a piece of the puzzle. That piece could be a new form of geological synthesis, a strange localized environmental condition, or, yes, the remnant signature of ancient biology. But regardless of which answer turns out to be true, the result is that Mars is more complex, more dynamic, and more mysterious than we assumed. The path forward is clear, though challenging. Either future experiments, including potential sample return missions supported by NASA, will broaden our understanding of Martian geochemistry and demonstrate that the planet can synthesize more complex organic matter than previously believed, or more detailed analyses, particularly isotopic measurements, will reveal signatures more consistent with biological origin. Isotopic analysis is the gold standard. Life prefers lighter isotopes of carbon. If we could measure the ratio of carbon-13 to carbon-12 in these specific hydrocarbon chains, we could get a much stronger indicator of their origin. Abiotic chemistry tends to leave a heavy isotopic fingerprint. Biology leaves a light one. Bringing samples back to Earth is the only way to do this with the precision required. The instruments we have on the rovers are miracles of engineering, but they cannot compete with the massive, room-sized mass spectrometers we have in laboratories on Earth. We need to touch these rocks. We need to pull them apart, atom by atom. This discovery in Gale Crater adds immense weight to the argument for the Mars sample return mission. It provides a specific, tangible target. Get us this rock, because it contains a chemical impossibility. For now, the ancient mudstone of Gale Crater carries a chemical signature that NASA must classify as anomalous within current models. It sits there as a challenge. It defies the radiation that should have erased it. It defies the meteorite models that cannot explain it. And it defies the simple geology that should not have produced it. It is a stubborn survivor, and it is precisely this anomaly that transforms the finding from another report of organics on Mars into one of the most compelling developments in contemporary planetary science. We are no longer just asking, is there carbon on Mars? We know there is. We are asking, why is this specific carbon arranged in this specific way? The sophistication of the question reflects the maturation of the science. We have moved from detection to forensics. We are investigating a crime scene billions of years old, trying to reconstruct the perpetrator from the fingerprints left on the weapon. This discovery reminds us that exploration is not just about visiting new places. It is about seeing new layers of complexity in the places we thought we knew. Gale Crater has been explored for over a decade, yet it still holds secrets that can upend our understanding. The red planet is not giving up its history easily. It demands that we look closer, think harder, and build better models. As we wait for further data and for the next generation of missions to launch, we settle into this new paradigm, a paradigm where Mars is chemically capable of surprises, a paradigm where the line between the geology of a dead world and the biology of a living one is thinner and blurrier than we ever imagined. The anomaly is real. The data is robust. The explanation is the prize that awaits the next great leap in planetary science. Thank you for joining me on this deep dive into the latest Martian mystery. If you value this kind of in-depth data-driven analysis, please like this video so more people can find it. And do not forget to subscribe. There are many more discoveries to cover, and we will be here to break them down, piece by piece. Until next time, keep looking up.